We are from RAS 1205A. For case study Chemist 260, we will present about development of a reverse phase Hplc method for determination of latent impurities of lenalidomide. Then this is all of our members: Arif Rene bin Yusuf, Hazim Zafran bin Zulkifli, Mimi Fiza Shamira binti Jamal, and Hana binti Alauddin. Okay, start with introduction. Lenalidomide is a medicine used to treat certain cancer like multiple myeloma and myelodysplastic syndrome. To make sure the drug works well and it's safe, it's very important to check its quality and ensure there are no harmful impurities, even in tiny amount. Organizations like International Council for Harmonization (ICH) set rules to identify, measure, and control these impurities. One of the best ways to test for impurities is using a method called high performance liquid chromatography (HPLC), especially a type called reverse phase HPLC (RP-HPLC). This method helps separate and measure the different substances in the drug, making sure it's pure and meets safety standards. The aim of this study is to develop and validate a robust reverse phase high performance liquid chromatography, or we call as a RP-HPLC method, specifically designed to detect and measure related impurities in lenalidomide. The focus is on optimizing the method to achieve high sensitivity, specificity, and reproducibility. This ensures the method is reliable and precise for routine quality control application in pharmaceutical production.
Now we move to the methodology part. The first part of the methodology is sample preparation. There was preparation of test solutions to analyze lenalidomide and its related impurities. Here how it is structured. The first one, we want to prepare test solution A. First, dissolve 10 mg of lenalidomide in a suitable diluent. Dilute to make a 1000 mg per liter solution. Test solution B. Dilute 1 ml of test solution A to 100 ml with the same diluent. Resulting concentration is 10 mg per liter. The last one is test solution C. Dilute 1 ml of test solution B to 10 ml with the diluent. Resulting concentration is 1 mg per liter. Next is a diluent use. Diluent consists of a mixture of mobile phase A and B in a 1:1 ratio, 50-50 V over V. Injection: 20 microliter of each prepared solution A, B, and C, along with a blank solution, was injected into the HPLC system. This is the second part of the methodology. Quantitative analysis. To determine the amount or concentration of lenalidomide and its impurities. The first quantitative analysis, which is peak area measurement. The peak area for each compound was measured from the chromatograms. Stress testing results. Lenalidomide purity was 99.91%. Calibration and comparison. Impurities in test solution A were quantified by comparing their peak areas to the reference peak. The purpose behind the quantitative analysis is first to ensure the method accurately measures impurity levels and detect degradation products. The second one is confirm the concentration of lenalidomide in various formulations. The second analysis is a qualitative analysis, is to identifying the type of nature of impurities and degradation products. The first qualitative analysis is retention time (RT). Lenalidomide show retention time when 17 minutes. Impurities appeared at 12 and 5 and 15 minutes under normal conditions. Second one is stress testing. New peaks observed after stress test indicate to degradation products. The third one is peak characteristic. The first one is resolution. Resolution meaning is the separation between peaks was measured to find distinct identification. The example for this analysis is resolution of 8 between impurities at 12 and 5 minutes. Symmetry and tailing factors is checked for the primary lane peak to ensure an accurate identification and quantification. The purpose we find qualitative analysis is to determine the presence of impurities or degradation product. And the second one is evaluate the robustness of lenalidomide under various stress conditions.
Next is for analysis results. In this figure 1, the chromatogram of the test solution as concentrated shows the low amounts of impurities is represented. Two impurities in retention times of 12 minutes and 50 minutes are detected, while the mean peak of Lena appears about 70 minutes. The resolution for the first impurity to the second and the second impurity to the main peak is 8.0 and 5.3. In this figure 4 shows that the oxidation stress caused by hydrogen peroxide H2O2 with given conditions decreased. Compared to the fresh test solution, the oxidation stress led to production of total 2.62% impurities. The first sharp peak at 4 minutes represents the existence of H2O2. The maximum absorbance of hydrogen peroxide occurs at about 220 nm, which is used to record all of our grams. For the figure 6 indicates the 10 ppm concentration of Lena as the middle dilute solution. It's detected by a high resolution, sharp 100 mV high peak with a tailing factor less than 1. As shown in the figure 7, this shows the last graph of this chromatogram. The 1 ppm concentration of Lena as the final dilute solution is clearly detected by a sharp 12 mV high peak with a tailing factor less than 1. This method could easily detect required low concentrations.
This figure highlights the effect of thermal stress on lenalidomide. The purity decreased significantly to 80.39% with 18.49% impurities generated after exposure to 90 degrees Celsius for 2 hours. The chromatogram shows well-separated peaks with high resolutions, confirming the method's robustness in analyzing thermal degradation product. Figures 6 and 7 demonstrate the sensitivity of the Hplc method. At 10 ppm, which is the figure 6, a sharp, high-resolution peak is observed with a tailing factor less than 1, indicating excellent chromatographic performance. Even at 1 ppm, which is the figure 7, the method detects lenalidomide with a clear, sharp peak, confirming its capability for trace-level impurity analysis. This highlights the method's reliability for both routine and sensitive applications. Now, let's talk about the discussion of the experiment. This study developed a robust reverse phase Hplc method to analyze lenalidomide impurities. The method showed high sensitivity, detecting concentrations as low as 1 ppm with sharp and symmetrical peaks. Stress tests revealed that lenalidomide is stable in acidic and oxidative conditions, with purities of 99.81% and 97.19% respectively, but it degraded significantly under basic and thermal stress, with purities dropping to 32.39% and 81.39%. This highlights the need to avoid basic and high temperature conditions during storage. Compared to previous methods, this approach is simpler, more cost-effective, and meets regulatory standards, making it ideal for routine pharmaceutical quality control. Overall, the method reliably evaluates both the impurity profile and stability of lenalidomide formulations.
In conclusion, the RP-HPLC method developed for detecting impurities in lenalidomide is reliable, sensitive, and specific. It was validated in accordance with ICH guidelines, demonstrating excellent accuracy, precise, and robustness. This method ensures the effective quality control in pharmaceutical manufacturing and helping to maintain the therapeutic efficacy of the drug and safeguard patients safely. The study emphasized the critical role of advanced analytical method in upholding high standards in pharmaceutical development and regulatory. Additionally, it suggests that this method could be applied to other drug formulation or impurities, offering potential for broader use in future research and quality control practice.